Thank you, Dr. Chan, and thank you everyone for coming to my presentation today. So over the summer and throughout the course of the year, I did a case study on the effects of a gluten-free diet on symptoms, the gut microbiome, and DNA methylation in vegetarian individuals with Crohn's disease. So to provide some background, Crohn's disease is a chronic inflammatory disease, autoimmune disorder, and inflammatory bowel disorder that can affect any portion of the digestive tract, but it's most common in the ileum. And Crohn's disease often presents itself with symptoms such as diarrhea, abdominal pain, rectal bleeding, weight loss, bloating, and fatigue. And when constant and persistent, these symptoms can severely impair quality of life. Therefore, researchers and physicians right now are implementing a drug called Remicade in order to help decrease inflammation and reduce symptoms in people with Crohn's disease. So the way Remicade works is it binds to an inflammatory cytokine called TNF alpha and prohibits this cytokine from driving inflammation in inflammatory biochemical pathways. But Remicade has shown to present itself with severe short-term and long-term side effects, ranging from fever, fatigue, and nausea to long-term side effects, as some studies have shown in liver failure and jaundice. So in order to kind of supplement the effects of Remicade and alleviate the side effects it might present, physicians and researchers are exploring various dietary interventions, one of them being a gluten-free diet, as it's a specially adapted diet and it's known to help people with gastrointestinal complaints such as Crohn's disease, and a gluten-free diet avoids all wheat consumption. So when changing the diet of anyone with Crohn's disease or without, we're bound to see changes to the gut microbiome, which is the community of microorganisms within our gut. And within that community, there are different bacterial families, genera, and species. And this bacterial families and genera that can change in abundance produce certain bioactive compounds, which can modify the structure of DNA called an epigenetic modification. And while the structure of DNA is changed, the nucleotide sequence remains the same. So the A, T, C, G, et cetera, will remain the same, but the structure of DNA will change as a whole. And that structure of DNA will thereby modify gene expression through a process known as DNA methylation. And DNA methylation is termed as a direct chemical modification to DNA that is involved in gene expression and regulation. So the way to think about DNA methylation is kind of if we're all in our house, we all turn on the lights based on the room that we're in. We're turning on like the lights to best suit our environment. So DNA methylation can serve as a way, and we're trying to turn on the genes that best benefit us and help us in terms of health and livelihood, and turn off the genes or supposedly silence the genes that aren't beneficial to us. So the way that it works is that if we remember to freshman biology, a gene is about 46,000 base pairs in terms of nucleotides, and the 100 to 300 base pairs upstream of the gene is what's termed as the promoter region of the gene. So the promoter region of the gene is where RNA polymerase is going to bind to, and it's going to transcribe the gene, which will then translate into a protein. But in DNA methylation, as we see in the top here, these methyl groups are going to sit on top of the promoter region of the gene. And when it sits on top of the promoter region, RNA polymerase can't bind to it as easily. And when it can't bind to the promoter, the gene isn't transcribed and that protein isn't made, which can help with inflammation and symptoms in that sense. So after doing this preliminary research, I formulated my research question in which how a gluten-free diet affects symptoms, the abundance of bacterial communities in the gut microbiome, and gene expression via DNA methylation. And I hypothesized that a gluten-free diet will help to reduce symptoms by promoting beneficial changes to bacterial composition, which can help to modify gene expression and downregulate inflammatory signaling pathways. So I first designed my study, and the major limitation I had with this project was that since it was a case study, I was confined to a small sample size, but I wanted to focus on vegetarian individuals since there's a gap in the literature to see how a gluten-free diet, in addition to a vegetarian diet, can influence things like gene expression and bacterial abundance. So I recruited one individual with Crohn's disease who is vegetarian to serve as my experimental, and my control was someone without Crohn's disease who is also vegetarian. And I put them both on a gluten-free diet for 35 days, sandwiched in between two gluten-heavy diets. And on days 0, 35, 70, and 105, a blood was drawn and a stool sample was taken in order to easily detect changes in bacterial abundance and gene expression as well. And a survey was maintained for all 105 days of the study to keep track of symptoms. And from there, after extracting the blood and taking the stool samples, I extracted the DNA from both the blood to analyze DNA methylation and the stool to analyze analyzed the gut microbiome, and I ran them through their respective sequencer, and I also analyzed the symptoms based on the survey. And from there, I went on to data analysis and answered my first research question on how a gluten-free diet can help to alleviate symptoms. And I found that there was a significant decline in symptoms post-gluten-free diet for the individual with Crohn's disease, as indicated by a McNamara statistical analysis test with a p-value less than 0.05. So before a gluten-free diet, we see that the individual with Crohn's disease is experiencing about 12 to 13 days of symptoms. Now, after a gluten-free diet, it drops to about 2 to 3, indicating a significant decline in symptoms. And now that I knew that there was a significant decline in inflammation and symptoms, I was curious to see what was happening under the hood, starting with the gut microbiome and bacterial abundance. So it answered my second research question of how this can influence inflammation. And I found that looking at this principal coordinate analysis, and the way I like to think about this principal coordinate analysis is that if we have a bunch of marbles in a bag and they're all red, and then at a certain time I take out the red marbles and I put in blue and pink one, the composition of that bag is changing, and it's changing in terms of the colors I'm putting in and taking out. And in the same way here, this principal coordinate analysis is reflecting a difference in composition at different time points. So what bacterial abundance is growing and what's getting taken away. So we can see here at time point 1 for the experimental, it's around here. After the first 35 days in a gluten-heavy diet, there's an upward tick. And then after a gluten-free diet, there's another upward tick, indicating a consistent change in the bacterial abundance that's growing and some that are going away as well. And then at time point 4, at the end of the washout phase, we see reflect back to baseline, indicating not too much of a difference in the gut microbiome composition between time points 1 and time points 4. And I noticed that these shifts were highly characterized by short-chain fatty acid producing bacterial genera. And so short-chain fatty acids, namely butyrate, acetate, and propionate, have been often given in trials done by the Cleveland Clinic and other trials across the world. And it's shown to have promise in decreasing inflammation with people with autoimmune disorders and inflammatory bowel disorders. And I noticed that while on a gluten-free diet, here, there was an increase in both butyrate and acetate producing bacterial genera for the individual with Crohn's disease. So the increase in this bacteria is doing what these supplements would do in these certain trials, and is helping to decrease inflammation and reduce symptoms while on a gluten-free diet. And I also noticed there was a decrease in lipopolysaccharide producing bacterial genera. And lipopolysaccharides have been often known to induce inflammation, trigger inflammatory pathways, and drive symptoms and increase symptoms and inflammation. But while there's a decrease on a gluten-free diet here, as indicated from 23.89 to before to 0.21 after, this helps to decrease inflammation and reduce symptoms, as we saw in the previous slide. And this helped me answer my third research question of how bacterial abundance can alter the structure of DNA and thereby influence gene expression. And I found that these short-chain fatty acids can alter the structure of chromatin by acting as a histone deacetylation inhibitor. So what is happening here is normally histones are going to sit atop the structure of DNA based on its charge and charge interaction. So when these histones sit on top of the DNA, it's condensing and tightening the structure of chromatin. But when these short-chain fatty acids come along, it allows for the removal of these histones from DNA, like removing speed bumps from a road. So it's really loosening the structure of DNA, and that loosened structure of DNA we see here in the second image here is helping to modify gene expression through DNA methylation. So as soon as that structure loosens a lot, it allows for an enzyme like a DNA methyltransferase to come in and methylate the promoter region of a gene, or for an enzyme like TET called 1011 translocation to come in and demethylate the promoter region of the gene, changing gene expression as a whole. And now, after seeing that this epigenetic modification made this change in gene expression possible, I went on to analyze 225 inflammatory genes based on DNA methylation. And I found that 168 out of 225 were differentially methylated, 54 being unique to the patient with Crohn's disease, 93 unique to the control, and 20 of them shared between them. So from there, I went on to aggregate whether these genes were hypermethylated or hypomethylated, so a lot of methylation compared to a little methylation, and I put them in what was known as a gene ontology network called Panther pathways. And Panther pathways produced 4 pathways: the interleukin, P38 MAPK, toll-like receptor, and apoptosis signaling pathways, indicating that these genes that I put into these pathways significantly help to drive these pathways forward. And from there, I found that there were 13 hypermethylated, so a lot of methylation in pro-inflammatory genes. So as a whole, these 13 pro-inflammatory genes that are being hypermethylated while on a gluten-free diet, their expression is slowly diminishing or supposedly being silenced. And as this is happening on a gluten-free diet, it's helping to decrease inflammation and reduce symptoms, which, as we saw in the previous slide with the first research question. And I specifically focused on two signaling molecules, IL-8 and IL-18, as these ligands have been known to drive inflammation in inflammatory pathways. And then from there, I went on to answer my last research question, was how did the abundance of bacterial genera and methylation patterns come together to influence biochemical and inflammatory pathways? So I started with what I like to call input pathways. So on the left-hand side here, we see the G protein coupled receptor. And what happens is these short chain fatty acids like butyrate and acetate can bind to the GPR43 receptor, inhibiting the transcription of inflammatory cytokines like IL-18 and IL-8, helping to reduce inflammation as we saw there. And in the toll-like receptor signaling pathway, lipopolysaccharides serve as an input into these pathway. But as we saw in the previous slide, there's a decrease in lipopolysaccharide production while on a gluten-free diet. So there's less input for these pathways. And with less input, the production of these inflammatory cytokines that this pathway produces is decreasing, as supported by the methylation pattern and helping to reduce inflammation as a whole. From then on, I went to my output pathways, the P38 MAPK pathway and NFKB pathway, in which I like to call these pathways positive feedback loops of inflammation. So what these pathways do is they take in an input with an inflammatory cytokine like IL-18 or IL-8 or TNF-alpha, and it's going to run through a series of steps to produce the same inflammatory cytokine, leading to this positive feedback loop of inflammation when significantly present. But now, while on a gluten-free diet, we see that there's a hypermethylation of these inflammatory cytokines. So there's less input for these pathways to take in. So they're producing less of the same inflammatory cytokine, helping to slow down this positive feedback loop of inflammation. And the last thing I found was the NFKB pathway produces what's known as FLIP, which is an anti-apoptotic factor. And apoptosis is termed as programmed cell death, and it's a pathway Crohn's patients often resist, leading to a harmful accumulation of inflammatory cells, macrophages, and T cells within the body. But during a gluten-free diet, as FLIP production diminishes, as the NFKB pathway production diminishes, it turns on these caspases. And these caspases then turn on the apoptosis signaling pathway, and these harmful cells can slowly die off. So overall, I concluded that the data supports my hypothesis and that a gluten-free diet by influencing gut microbiome composition can conceivably influence the structure of DNA, thereby changing DNA methylation patterns and helping to silence inflammatory biochemical pathways, altogether helping to reduce symptoms, decrease inflammation, and reduce drug reliance. So my takeaways and advice for future researchers were to be creative and think outside the box in terms of coming up with this project, to enjoy the process rather than focusing on the results, and to be willing to practice endurance, responsibility, and organization. And last but not least, my acknowledgements. So first off, I want to thank Ilani School, the Ilani Wet Lab, for allowing me to use their facilities over the summer and throughout this year to conduct this research. Papa Jack for funding the research I did over the summer through the K Fellowship. Ms. Kobayashi, who has been a stalwart supporter of my research since day one, and always somebody I can talk to and have a laugh with. Dr. Chan, who I remember coming into her office as a confused junior, and now I'm presenting here, and she's taught me so much about not only science, but how to present research in terms of written expression and spoken expression as well. Dr. Perez David, who has taught me so much about genetics, the epigenome, gut bacteria, and so much more as well, not including science, but he knows what I'm talking about. But I guess you get what you asked for. And last but not least, Mr. Hill, who couldn't be here today, his patience throughout this entire process has been extraordinary in terms of helping me with devising my research plan, doing lab work with me, and helping me read, write, and present. And last but not least, to my family and friends for their never-ending love, support, and belief throughout this entire process. Thank you, and I'm ready for questions.